Welcome to a new episode of P Cubed. Actually, this is the second episode, and I'm honored to have our guest, Dr. Roberto Salgado. Roberto is an international expert on immuno-oncology biomarkers and developing guidelines, and that's why I wanted him to be here. He practiced in ZAS hospitals in Antwerp, Belgium, but he also worked internationally. So he's an honorary research associate at the Peter MacCallum Cancer Centre in Melbourne, Australia, and Francis Crick Institute in the research division in UK. I don't know how you do it with all these disparate locations, but clearly you've done an amazing job. Most importantly, for the sake of our podcast and our audience today, he's a co-chair together with Shireen Loy of the International Immuno-Oncology Biomarker Working Group. And what I love about that role is this is a working group of more than 900 pathologists over 60 countries. Amazing, Roberto. Congrats. That they're all putting their heads together on trying to see, now that immuno-oncology is very important, how do we develop guidelines for pathologists to do the best job on that? I'll be remiss if I don't mention that Roberto and I meet regularly at the WHO. I had the honor to meet with him as a member of the editorial board on breast cancer. And today, the conversation will center about an editorial that him and Shireen wrote discussing a new study that just came in Cancer Cell on use of antibody-drug conjugates in breast cancer. So thank you, Roberto, for joining me, and I'm really honored you're here.
No, thank you very much, George. It's the honor is completely mine. I really love the opportunity to engage with my fellow pathologists through this medium. Thank you very much for the invitation.
It's obvious. Now you're going to be almost like being in a Hollywood movie. You're going to be all over the globe, and people will see
virtual
through our podcast. They're going to see your beautiful beard and hair. I'm jealous from the hair because, as you know, I don't have much remaining. I'm working on it. So
it's lessening also from my side.
It's part of maturity. So let's go back to the topic at hand. If you start, since the study that's going to launch the conversation is on ADC, a little bit about ADC and breast cancer from your experience. Just give us a little bit mini symposium.
Well, indeed, it's actually a very fascinating topic for a variety of reasons, and I will try to detail them very briefly. The first one is the antibody-drug conjugates. How do they work? It's very simple. They use HER2 on the membrane as an anchor, as a ploy. So they bind, and then they get like a torpedo into the cancer cell. The cancer cell explodes, and all the chemo gets out. That's how it works. So the difficulty is HER2 as such a biomarker, yes or no? And now the entities that treated with these drugs who respond very well, are they separate entities, or is it just a mechanistic thing about HER2 being on the cancer cell membranes, serving as a ploy and nothing else? So that's one element. Is it a biomarker, HER2 expression? Yes or no? And that's important when we discuss the ASCO-CAP guidelines on the HER2 low and the ultra low. If we don't consider that this is a biomarker, then the reasoning becomes a bit different. Why? Because then only quantity matters. When we tend to think about a biomarker, it means that the cancer cell is dependent on the biomarker for whatever, for its growth, its proliferation, its invasion. But now we're talking about the biomarker is there, or HER2 is there, just to serve as a ploy. So now the question comes, based on a variety of prospective phase 3 trials called the Destiny-04 and the Destiny-06, which the Destiny-04 has introduced the concept of HER2 low, and the 06 Destiny trial has introduced the concept of HER2 ultra low. Now, what does it mean? What does it mean? It's very simple. The lower the HER2 expression on the membranes, the more you get to the HER2 ultra-low. What does HER2 ultra-low actually means? We know in the classical ASCO-CAP guidelines that you have HER2 zero, which is actually two entities. HER2 without any expression and HER2 with a little bit of expression, up to 10% of tumor cell. This means that one positive tumor cell, is called HER2 ultra-low. We as pathologists, we know the more we look, probably we will find that single positive tumor cell. And again, thinking, is HER2 then a biomarker? Should we really assess HER2 as a biomarker, yes or no? We can discuss this later on during the podcast. Now, the difficulty for daily practice is, first of all, these drugs are extremely expensive, but they work quite well. But they cause life-threatening toxicities, certainly in the lungs, interstitial lung disease, and a variety of other complications. So it is crucial that we find patients who may probably not respond very well to this treatment. So we shouldn't be giving them this treatment because the toxicities can be extremely severe. And sometimes patients die due to toxicities and not due to the cancer.
So you really want to give it to people who would respond. That's why it's crucial to have the predictive.
Absolutely.
And I love the view of, is this a biomarker that is helping the tumor or is just a docking station now?
Exactly.
Sounds like we're using it now as a docking station, because if it's in one cell, clearly that's not that critical anymore for the tumor. It's critical for us to target the tumor. I love that thinking.
Absolutely. Yeah, and it goes even further, meaning we all know as pathologists that practically every breast cancer has HER2.
Wow. It's just how low do you want to go?
So how low do you want to go? So that's the major concept. Now the question is, there has been a tremendous variety of research ongoing, and none of them has found one suitable predictive factor. But, and here comes the trick, and here comes the trial. It's a phase 2. We can discuss about the phase 2 trials. Is that sufficient to change practice? But phase 2 trials can be very informative to inform us on the biology. So the Fascinate trial is a phase 2 trial in HER2 positive disease. Now, let's forget a little bit for the moment the HER2 low and ultra-low story. Let's consider antibody-drug conjugates in HER2 positive disease. Okay, that's a concept. And then we will combine both of them together. A Chinese group from Funan Cass University, it's a tremendously powerful and very influential group. They have developed a phase 2 trial with a single agent, so without chemo and an antibody conjugate, just the antibody-drug conjugate. They gave it to 60 patients with HER2 positive disease. And what did they saw? That's the manuscript that they published in Cancer Cell a few months ago.
I just want to interject. The lead author is Ding Ma, and this was published probably last month or the month before in Cancer Cell with the editorial that is authored by you and Shireen.
So what did they find? And that's very interesting. They find, and I will tell the results immediately, and then how did they come to these results? Basically, they saw, first of all, that in the HER2 positive, hormone receptor negative population, the immune system is actually a very powerful predictor for response. That's one. In the hormone receptor positive, HER2 positive population, the immune system wasn't actually that important. What was important there is, let's call it the architecture of the cancer, how the cancer looks like. And we know when we look through an H&E, we see a variety of patterns. But we never considered formally that those patterns that we see may tell us something about, will the drug respond or will the drug act on the cancer, yes or no? So this group found that in hormone receptor positive disease, the closer the cancer cells are packed, the less
the ones that
the
are
drug
expressing HER2, right?
All HER2
cells
positive.
Expressing HER2.
All cells are expressing HER2, HER2 positive, hormone receptor positive. the closer the cancer cells are packed, the more solid the growth pattern is, the less the drug works.
Wow.
And the less packed, the less closer, so the more disaggregated, the better the drug works. Now,
so patterns does matter.
Absolutely.
And you can do that with AI.
Architecture, exactly. And that's the point, because I can score the TILs with a microscope. You can do it with AI. But architecture is something that we see with a microscope. But don't ask me to quantify it. Don't ask me to put a number on it. And don't ask certainly me as a pathologist to give it a number and integrate it in a predictive model. Because what have these guys done? These guys have they have combined the clinical variables, okay, the pathological variables, and then they did some spatial profiling, which was validated using DNA and RNA sequencing data, and combined the spatial profiling with immunohistochemistry and the spatial profiling on an H&E. They put it all together in a predictive, what they call a predictive model. And that model was actually very predictive in predicting which are the patients that will respond to antibody-drug conjugates. Meaning, they saw that the stronger the immune system, and you can measure the immune system with an NHNE just looking through the microscope or using TILs, or you can measure it with more complicated immune metrics, what they have done, finding always the same thing. But architecture is something that we can't measure, and that's an important lesson. When I read the manuscript, because I was one of the reviewers, I was wondering, would I be able to quantify a pattern for clinical use? And my answer, to be honest, is no. I can score the TILs very easily. I can do a CD8. You can do genomics for immune system. Practically assessing the immune system is important, whatever way you do it. But assessing a pattern, for that you need AI. And what they did, they combined everything together. Now, what they are now doing as a next step is validating this predictive model in a subsequent trial. And that's important how we want to progress, because a phase 2 trial is informative. And that's it. So it doesn't change conceptually, it doesn't change clinical practice. So it doesn't mean that we have to start immediately implementing this predictive model in our daily practice.
So validation is critical, and you just beautifully described how this study took, and by the way, the antibody-drug conjugate name is HRA 1811. So it's just an ADC that home on HER2. And again, I want to emphasize this was HER2 positive in this trial. But you mentioned this multimodal approach to AI models, and multimodal meaning H&E, looking at lymphocytes that are stained, looking at HER2 cells, tumor cells that are stained, all this with AI, but also the clinical, the pathologic, and the DNA and RNA data. This is the power of AI, but our piece, the pathologist, is important, and we want to get to a point where can we start having an algorithm to provide our piece, right? And say this is dense, this is architecturally heterogeneous, and architecture, whether it's dense versus spread out, of HER2 positive cells matter, and can you put a number on it, maybe with an AI model? That's very exciting, and it's really germane to why this podcast. I always want to focus on studies like that bring the future power of AI, of genomics, and this combine both. Go ahead. I interrupted you. You wanted?
No.
I think it's a good point now. Beside the study, and I refer you to the figure for the audience, to the figure in the editorial, beautiful figure, simplifying, describing exactly what Roberto shared with you about the study from our point of view as pathologists. Can you say a few words about the HER2 low and ultra low,
yes,
the ADC landscape, and anything from our side that we need to know as pathologists, or in any solid tumor, since I know you do other than breast?
Absolutely. the first element is how should we measure HER2? So we have the Destiny 04 has introduced HER2 low in the public domain. The Destiny 06 trial introduced the concept of HER2 ultra low. So what we don't know, based on these two trials, is whether no expression, 0, by the pathologist predicts benefit. This is being tested in an additional prospective trial called the Destiny 15 trial, which will include patients with score 0, no expression at all. And then we will know. But, and here comes the thing, we have been trained, all of us, by the concept of clinical utility and clinical validity. So you need prospective randomized design. Now let's think critically as pathologists. And here comes the nuance about how we translate clinical trial findings to daily practice. The Destiny 06, the analysis for HER2 ultra low in that particular trial, it was a magnificent trial, by the way. It was an exploratory subgroup analysis, and it wasn't an integral biomarker in the trial. Now, think again. It's a research objective, it's an exploratory biomarker, and it was not integral to the trial. And yet, and people jumped at it because it's a drug, you can give a drug, and you can find patients who respond. So imagine, that's what I tell my peers, is imagine you have an academic group who does a prospective phase 3 trial, and they do an exploratory analysis with a particular biomarker, let's call it TILS or whatever. And then they go to the FDA, they say, we want this biomarker to be approved as a biomarker, we want the assay to be approved as a companion diagnostic, and we want the drug to be approved. The classical comment also by academics would be, but you haven't done a prospective randomized clinical trial. Now here comes Big Pharma going to the FDA with the same thing. And they get a companion diagnostic, they get a drug approved. And now here comes the nuance. Daiki is completely right. Why? Because the biology is the docking station. So it is completely rational to treat patients with HER2 ultra low with this drug. Because what the drug does, it binds HER2 and it gets into the cancer cell. So it is perfect
for the drug. And that's the only thing we need for the drug.
That's the only thing.
Yeah, we'll see if the complete zero, no HER2 expression at all will work. But then how are you going to argue for that, in terms of using docking if it's not on the surface of cell? It could be that it is in a way, but it's not being labeled. And so, but we'll deal with that on another podcast when that trial, you said is it's Destiny 15, right?
Destiny 15. And you know what would solve a lot of things is coming back to the AI spectrum in the digital pathology space. What I firmly believe, based on evidence, is that we need clinical trials to validate these assays. So what we see in the literature, in all journals, is a lot of wonderful science based on large retrospective datasets or publicly available datasets who are very informative to the community, but they are not driving the change in daily practice. What is driving the change in daily practice is the validation clinically in large randomized clinical trials that have been finalized. To give you a very concrete example, we have just finalized with Friends of Cancer Research a comparison of up to 10 different HER2 assays using AI. And we did see variability between the same variability that we see between pathologists reading AI. And we see the same artifacts causing this variability. So nothing that surprising, to be honest. The question now is, does it matter? Does it matter if you have one AI assay who scores a 0 and the other one who scores 1+? Does it matter when you have one AI assay who scores ultra low and the other one scores HER2 low? The only way in which we will know is to test these assays in trials that have been finalized. And industry, and I work a lot with industry and I love them really, they are extremely reluctant to use their samples for this type of comparisons for a variety of reasons. FDA is not willing to do them, and they're scared for the label and this and that, and perfectly understandable. But we need to go beyond these doubts. So as a community, and I will use this podcast to spread the message, we need to get clinical trials available for validation of all those wonderful digital AI assays that we are developing. Otherwise, we will not be able to bring that level of evidence to the community.
Wonderful. This has been very enjoyable, and it sounds like we need another episode to talk further about AI and immuno-oncology with you, and I'm sure the opportunity will come. I'm really grateful for the great conversation, and I wish you and the working group the best.
Thank you very much, Zeus. Thank you very much for the invitation. And I would love to continue to engage with you and all your activities. Thank you very much.
Thank you. It's such an honor. For our audience, I hope you enjoyed it. Please listen to us. And like I just mentioned, now we have a vodcast. So on YouTube, you can see the episode and see the successful pathologist, Roberto Salgado, on video too. And cheer till next episode.